Assalam- Assalamu alaikum. My name is Jannat from Department BS Biotechnology. My presentation topic is L-Phenylalanine. So let's get started. Let's have a look on the content. First of all, there is introduction, then structure, physical and chemical properties, computed properties, preparation methods, applications, summary, and then reference. Introduction. The production of L-phenylalanine by transamination was first explored about 30 years ago. Transamination is a chemical reaction that transfers an amino group, keto acid, to form new amino acid. Keto acids are also called as exoacids. L-phenylalanine, which have the morphology of spherolytic needles and amino group constituent responsible for brain nutrition, is crystallized in silica gel of pH 6 by single diffusion method of 30 minutes. The ground crystals were characterized by X-rays, powder diffraction, Fourier transforms, infrared spectrography. Amination and de-racemization. Amination, which is introduced in organic molecules, whereas de-racemization is de-racemization mechanical. Its molecular formula is C9H11NO2. The synonyms of L-phenylalanine are phenylalanine 63912, S-phenylalanine, 3-phenylalanine, H-PHEOH, beta-phenylalanine, 3-phenylalanine, antibiotic FN1636. Its molecular weight is 165.4 gram per mole. There is 3D structure and chemical structure of L-phenylalanine. In its chemical structure, benzene ring is attached with the amino alcoholic group and sharing double bond with oxygen. Physical and chemical properties. Physical description. It is an odorless white crystalline powder. It is slightly bitter in taste and pH 5.4 to 6. It is colorless or white plate-like crystals or dry powder. It is in the form of solid. Color form. Prism form water. Odor. It tastes bitter. Density 1.34 gram per centimeter cube at 20 degrees Celsius. Boiling point 563 Fahrenheit at 760 millimeter of mercury. Its melting point is 541 Fahrenheit. Solubility 10 to 50 mg. It is insoluble in ethanol C2H5OH, ethyl ether C2H5O, benzene acid C7H6O2. It is very slightly soluble in methanol and ethanol. Its vapor pressure is 1.76 multiplied 10 to minus 8 millimeter Hg at 25 degrees Celsius. Log P minus 1.38. Optical rotation. Specific optical rotation is minus 35.1 degree at 20 degrees Celsius. Decomposition. When heated to decomposition, it emits toxic fumes of nitrogen oxides. Dissociation constant. Acidic pK 2.2, basic pK 9.08. Computed properties. Property name and property value. Its molecular weight is 165.19, X log P3 minus 1.9. Hydrogen bond donor count is equal to 2. Hydrogen bond receptor count 3. Statable bond count 3. Monoisotopic mass, and so on. The concentration of L-phenylalanine. Preparation method of L-phenylalanine. The preparation method of L-phenylalanine. Preparation of N-salicylol L-phenylalanine. It was prepared by simple two-steps process from phenylalanine benzyl ester and acetyl salicylyl chloride, followed by hydrolysis and catalytic hydrogenation in an overall yield of 85%. Hydrolysis is a chemical reaction of the interaction of chemicals with water, leading to the decomposition of both the substance and water. Catalytic hydrogenation is the treatment with hydrogen in the presence of catalysts such as nickel, platinum, palladium. Enzymatic production of L-phenylalanine through transaminase reaction. To develop an enzymatic process for L-phenylalanine production through transaminase reaction, a cytobacter fluendice strain is an excellent producer of the enzyme. The cell of this bacterium rapidly and efficiently formed L-phenylalanine from phenylpyruvic acid, and an amino acid donor with L-phenylalanine production reached 30 mg per ml in 1 hour and 89 mg/ml in 24-hour incubation. The conversion the conversation yield to form L-phenylalanine from phenylpyruvic acid was 50 to 80%. With L-aspartic acid as amino acid donor, the rate of L-phenylalanine production is lower, but the conversion yield was much higher. With L-phenylalanine acid as the amino acid donor, oxalic acid putative reaction product appeared in small amount. Instead, a much large amount of pyruvic acid, a well-known decarboxylation product of oxalic acid, appeared. From this fact, it was concluded that the reaction, which should be reversible, inclines almost completely toward L-phenylalanine formation when L-aspartic acid is eliminated through decarboxylation to form pyruvic acid and other metabolites. The Citrobacter strain had a high aspartate activity for pyruvic acid and other metabolites. Applications. It is used in essential amino acids for humans and in most livestock. Livestock are farm animals except the poultry animals. It is used in feed, food, additives, taste, and aroma enhancer. Additives are a substance added to something in small quantity to preserve it, like monosodium glutamate. It is used as drug, dietary supplements, nutraceuticals, and ingredients in cosmetics. Nutraceuticals are products which, other than nutrition, are used as medicine. L-phenylalanine mustard in management of primary breast cancer. Prolonged L-phenylalanine are used in the management of patients with primary breast cancer, and pathologically positive axillary nodes were evaluated by prospective randomized clinical trial. Axillary means armpit, and axillary nodes are the first place where breast cancer spread first. Because of the effectiveness of chemotherapeutic agents used singly or in the combination in the management of patients with advanced breast cancer, treatment of women with a lesser burden is the high priority. Thus, L-phenyl, uh, L-phenylalanine mustard has demonstrated to be effective in this disease. Results were achieved with minimal undesirable side effects. Phenylalanine ammonia lyase, PEL, which catalyzes the biotransformation of L-phenylalanine to transaminase acid and ammonia. PEL has gained considerable significance in several clinical, industrial, and biotechnological applications. The enzyme's naturally ability to break down L-phenylalanine makes PEL a reliable treatment of genetic condition phenylketonuria. It is phenylketonuria. It is a rare inherited disorder that causes an amino acid called phenylamine to build up in the body. Applications. Currently, L-phenylalanine is obtained by chemical, enzymatic, or microbial processes. In recent years, there has been increased interest in producing L-phenylalanine through microbial fermentation, especially by metabolically engineered strains of E. coli, which has the high growth rate and well-defined physiological characteristics. It is only form of phenylalanine found in proteins. Major dietary sources of L-phenylalanine include meat, fish, egg, cheese, and milk. It is found naturally in the breast milk of mammals. It is used in the manufacture of food and drink products and sold as a nutritional supplement for its analgesic and anti-depression effects. It is a direct precursor to the neuromodulator phenylalanine, a commonly used dietary supplement. It's an essential amino acid. It is not synthesized in human and other animals who must ingest phenylalanine or phenylalanine containing proteins. Summary. Phenylalanine is an essential amino acid. It means that the human body is unable to synthesize its own phenylalanine. People must instead get it from dietary sources. In general, food that are high in protein are also rich in phenylalanine. It is involved in the production of neurotransmitters, norepinephrine and dopamine, which are essential for proper functioning of brain and nervous system. It also helps with the production of the pigment melanin, which gives color to the skin, hair, and eyes.